first polar body. And all of that is going to be surrounded by the protein coat, which would be the zona pellucida. And then we would have the corona radiata. And additional follicular granulosa cells around that. So that's what is going to be released when the mature antral follicle ruptures and the fimbrium moves close to the ovary and takes it up. So what's the chromosomal pattern? Are these two cells going to have the same chromosomal pattern?
Yes.
Yes. And what will it be? No. How many meiotic divisions have we had? Just one. So our primary oocyte prior to ovulation was 2 parentheses 2n. And then just prior to ovulation, we had our first meiotic division, creating the secondary oocyte, which is 2 parentheses n. Remember, this is the first time we see a haploid cell, but we still have the chromosomes that have been replicated. So if this is egg, it's going to have the haploid XX, and the polar body over here would have the other haploid XX that came from the mother or the father of this particular individual. And so the secondary oocyte is 2n, and the first polar body is also 2n, but 2 parentheses in. So indicating it's a haploid cell. All right? So this is what enters into the ovary, the oviduct. And as it progresses through, remember, unless it gets fertilized, it never completes that second meiotic division. All right? So now we introduce the sperm. And as the sperm move up through the uterine cavity, they will pool here in the isthmus, and then waves of several hundred sperm at a time will move into the oviduct. And so it's not millions of sperm that are at one oocyte at a time, but several hundreds. And their acrosomal enzymes, some of them will be released earlier and will digest through this cluster of cells that is around the egg, creating a channel, and then the sperm, the first spermatozoon, to reach the egg will undergo an acrosome reaction, providing the receptors to bind to that zona pellucida. So it's a series of events that needs to occur to provide for the ability to undergo the acrosome reaction. Now, we talked about changes to the sperm that occurred in the tail of the epididymis, and that was maturation of the enzymes to allow the flagellum to beat, all right, and some destabilization of the membrane allowing the acrosome reaction, but the acrosome reaction doesn't take place yet. So, within the female reproductive tract, and specifically within the oviduct, a series of events called capacitation takes place. And that's further destabilization of the plasma membrane around the head of the sperm, allowing the acrosome reaction. So without capacitation, the acrosome reaction won't occur. All right? Now, it used to be included with capacitation. There is an increase in the head wiggling, but that's now seen as a separate event. So capacitation is just further destabilization of plasma and acrosomal membranes. Right, when I say plasma, I'm talking about the plasma membrane of a cell, because because even the acrosomal membrane is a plasma membrane, all right? But I mean the outer membrane. Which will allow the acrosome reaction to occur. Okay, so that takes place within the oviduct, and it requires the presence of progesterone. In fact, remember that lab at UC Davis I told you that I worked at, where we did the sperm? That was one of the first, if not the first lab to discover the requirement of progesterone needed for this capacitation to occur. So let me see if I can, I did do it for the morning class. Let's see if I can pull it up really quickly for you guys.
So if the female has low estrogen or progesterone levels,
Or if these cells have insufficient receptors
fertilization
for them, then it won't occur.
Professor, this is happening in the sperm?
That's happening to the plasma membrane of the sperm. Yeah. So capacitation requires the milieu or the environment of the female reproductive tract, but it's changes in the sperm. So if we're doing in vitro fertilization, that same milieu and concentration of chemicals has to be present in the petri dish for the sperm to be capacitated. All right, so this video right here is showing a scanning electron micrography of the acrosome reaction, which I will show on the screen, and then the downloads. While it's downloading, let me kind of diagram what's going to happen. So what you're looking at is the head of the sperm, and you can see that there is this crinkling effect. That's the fusion of the outer plasma membrane with the outer acrosomal membrane and its vesiculation. So before I show that again, let me diagram what's happened. So this is acrosome reaction. This is going to occur to some of the sperm as they're making their way through the follicular or granulosa cells, and then the actual binding of the sperm to the egg triggers it in the key sperm that's going to actually, if it occurs early, then those sperm don't fertilize the egg, all right? It's when it will occur to the sperm that binds to the zona pellucida for the sperm that's actually going to fertilize the egg. So here's the plasma membrane. And the nucleus. And then remember the acrosome fits like a stocking cap, pulled down over the nucleus. And that destabilization that occurs is a breakdown of these two membranes. So this membrane here is known as the outer acrosomal membrane. And what we'll see again when we watch that is a breakdown between these two membranes, and then they actually fuse together to create little sacs or vesicles. And these will close. All right? So those are little sacs, and they're not connected to anything anymore, just themselves, right? And so those will get washed away, and this is what we have left for the sperm that actually binds to the zona pellucida. Is what actually binds are receptors on this inner acrosomal membrane. That's what actually binds to the zona pellucida. That actually is species specific, all right? So The receptors on this membrane for a human sperm won't bind to the zona pellucida of a hamster or a horse. Once the zona pellucida is, when this binds, I'm sorry, I did it reverse. When this binds, it's this, when this binds to the zona pellucida, let me redraw that here because I said it backwards. So in the sperm that's going to fertilize, this outer plasma membrane will bind to proteins on the zona pellucida. These proteins are known as the ZP3 protein or zona pellucida protein. And the receptors for that are on this outer plasma membrane. That's the species specific. So that will recognize specific proteins on the zona pellucida. Once the acrosome reaction occurs, this is non-species specific. So this inner plasma membrane will bind to the egg membrane, the plasma membrane around the secondary oocyte of any species. All right, it just has to get through that zona pellucida first, and getting through the zona pellucida is what is species specific. That's kind of just for free. So let's watch this video again. So what you're seeing at first is the outer plasma membrane of the sperm, and then it's starting to look like it's buckling up or wrinkle, and that's when the outer plasma membrane and the outer acrosomal membrane are fusing together, and then they'll disappear and you'll see just the inner acrosomal membrane. So the outer one is clustering and fusing with the outer acrosomal membrane. And now you can start to see the inner acrosomal membrane as that's fusing and getting flushed away. And so we're left with this edge, which is this edge right here. So after the acrosomally reacted sperm is going to look like this. Okay? And it's this inner acrosomal membrane that will bind to the egg itself. So that's what we're going to look at. And it's shown, this process is shown on the bottom of page 120 of your lecture notes. It's showing it for a sperm. I'm sorry, it's showing it for a sea urchin. So several of these sperm coming in will react early and release the digestive enzymes to separate these follicular cells and get to the zona pellucida. And then one of them will have receptors for the ZP3, which is a protein of the zona pellucida that triggers its acrosomal reaction. And you can see by these little edges there, the outer plasma membrane has been lost along with the outer acrosomal membrane. Those enzymes will digest through the zona pellucida, and then the sperm comes in kind of sideways and binds to receptors on molecules on the egg membrane itself. All right? So the ZP3 is a zona pellucida protein, and then it's going to bind to the egg protein. All right? So in this first slide up here, this is what I drew on the board earlier. So we have our secondary oocyte. Notice that it's ready to start to divide. Okay, it's primed for the second meiotic division. Here's the first polar body. And both of those are the two parentheses in. So this actual separation into two cells, the otid and the second polar body, occurs with the penetration binding of the sperm. So here comes the sperm, all right? Here's what we talked about already with the acrosome reaction. Comes in for that sideways landing. And that's where it binds to the surface of the egg. Now I've enlarged this. So that's the inner acrosomal membrane. And then our DNA will be in there. So this will be the nucleus.
Which binding is
So this is the secondary oocyte, and this protein that it's binding to is integrin alpha 6 beta 1 protein. So the ZP3 is the zona pellucida, and the integrin alpha 6 beta 1 is associated with the egg.
And which one is species specific, the egg?
The ZP3 receptor binding. So getting through the zona pellucida is species specific.
So how do they, like, mate a mule with a donkey or whatever?
So what you can,
Crossbreed.
that will occur, so sometimes we can get cross species, but then that animal is sterile, so it won't continue, okay? So the protein, the integrin alpha 6 beta 1 is the protein on the secondary oocyte, and the inner acrosomal membrane has a receptor for that, okay? So that's the interaction between the sperm and the secondary oocyte, and that triggers the fusion of these two membranes and the insertion of the head of the sperm into the egg. The tail, microtubules are left out. So the head of the sperm, the DNA, and a couple of centrioles will go in because the egg doesn't have those centrioles. So now what we have then is a secondary oocyte with the head of the sperm over here. It actually tends to do that on the opposite side of the egg where the egg's chromosomal material is. And that binding triggers the egg to undergo the second meiotic division. So as this is moving in, that binding here is going to trigger the cell to undergo secondary meiotic division, and we're going to form the second polar body. This was our first polar body. Still inside the zona pellucida. All right, so now our cell is an ootid. In, and our second polar body is in. Our first polar body may divide again. If it doesn't, then it's still going to be 2 parentheses 2n. All right, so in this image, here's the sperm. The oocyte has divided. Here's the ootid. Has the sperm head in it, but the ootid nucleus is not yet joined with that. So what we have is just the sperm nucleus inside the ootid for 2 separate genetic components. First polar body and second polar body. Okay. Good. Then the sperm nucleus migrates towards the ootid nucleus. They're called pronuclei. And the two move together. All right. And associate in pair up again. So if the sperm nucleus is carrying a Y chromosome, it pairs up with the X chromosome and so on, and we undergo replication in our first cell division. But that fertilized egg, once the chromosomes move together, is now called a zygote. Okay? So within the zona pellucida, we have a new cell with new chromosomal pairings, a new 2N, but a zygote is diploid. Once the male chromosomes and the egg chromosomes, the sperm chromosomes and the egg chromosomes come together as a zygote, that is a new diploid or 2N cell. Every division that occurs, unless we're talking about the gonadocytes again, is going to be a mitotic division, producing 2N cells. All right? So if we look at our models over here, here is the secondary oocyte. The yellow would be the oocyte with its nucleus. This would be 2 parentheses N. And the zona pellucida and the corona radiata and all the other follicular cells around it. Same thing without the corona radiata cells shown here. With the first polar body. So this would still be a secondary oocyte. 2 parentheses N. First polar body. 2 parentheses N. Okay. Now, the sperm comes along. This isn't showing the first polar body, but it would have a first polar body still. And 2 parentheses N. And the secondary oocyte is 2 parentheses N. Now that it's been fertilized by the sperm, it's going to undergo mitosis. I'm sorry, it's going to complete the second meiotic division and form an ootid and a second polar body. So we have an ootid that is now in, a second polar body that is in, and the first polar body that is still 2 parentheses in. All of it still surrounded by the zona pellucida. Okay? Where is this taking place? In the oviduct, in the ampulla of the oviduct. Okay, good. Now, we'll come back to the divisions a little bit later. Let's go back to the time of fertilization. And I'm on page 122. Talking about blocks of polysperm, because we don't want 3 or 2 sperm. If we had 2 sperm in there, donating its in, what would be the chromosomal pattern of the new cell?
XY1.
Well, just the chromosomal pattern would be 3N. So we'd have the N of the egg, the otid, the N of the first sperm, and the N of the second sperm. So it would be a 3N cell, and that's not viable. Okay, it might divide a couple of times, but it's not going to go and even implant into the uterus.
So where do maternal twins come from?
Okay, so twinning, and we'll get back to blocks of polysperm in a moment. If you have twins that are not identical, that is 2 separate eggs fertilized by 2 separate sperm. Okay? So it may be 2 eggs from 1 ovary, or it may be that both ovaries ovulated at the same time. If you have identical twins, then it is 1 egg fertilized by 1 sperm that has gone through 2-cell, 4-cell, 8-cell division. Sometime in there, the cells that were dividing separated and each continued to divide and form 2 separate balls of cells. The farther down those series of divisions you go, the closer you get to Siamese twins, where the separation was so late that the developing embryos have parts of it still fused together and maybe organs that were shared, not just skin. Okay, so that depends on the timing of when that separation occurred. Now, this is for free, but you can have the polar body, the second polar body might also get fertilized, or the first polar body might also get fertilized. By itself, it's not viable, because it doesn't have enough cytoplasm to support all of those cell divisions. But it can fuse with the larger ootid, and it would be a different chromosomal pattern, right? Because it was random division in the crossing over, and has different of those chromosomes. If they fuse, then we have something that's called a chimera. So the 2 individuals fused together. And the cells would have, part of the cells, patchy cells, would have different sets of chromosomes. So there was a CSI Miami show where they had the DNA of the individual. They knew they did, but it didn't match the individual when they went back to test it. And that's because they'd taken cheek cells and were matching them to sperm cells. The cheek cells were from a chimera. Even though they came from the same body, they had different chromosomes because they were from an early, like the polar body that fused. So they had different chromosomes than the sperm cells, which came from the other egg cell. Weird. It's like two people fused together. And
But not Siamese twins?
Not Siamese twins. Siamese twins would have the same. So this would be, Siamese twins would have, say, the zygote. So here's all the chromosomes from the egg, all the chromosomes from the sperm fuse together and create one cell, all right? And then that cell would, those series of cells would separate, and over here they would continue to divide, but it's through mitosis, so they would all have the same combination of 2 N. So they would be cloned, and they would have the same genetic component. Okay? With the chimera, and the fusion, say, of the, let's say that the cell got fertilized by 2 separate sperm and then fused together, you would have cells that would be in the same population. Some of them would have the cells of the egg and 1 sperm. The others would have cells of the egg and a different sperm. And then those would fuse, and some would become part of the skin, and others would become part of the skin, but one part of the skin would be from 1 cluster of cells, and the other part of the skin would be from another cluster of cells. You can see it in animals. Sometimes the fur will be different colors. You can even see it in humans, and if it's the different pigmentation, there'll be different patches of skin, different colors in that chimera. If it's in an organ, you can't tell. You just have to do a genetic test of each component. So the chimera is 2 different cell genetics fusing together and each multiplying but forming one body, one organism. Okay? I kind of got off track there, but it's a really interesting forensics component where a person has 2 different sets of DNA. So the fast block, back to the sperm, binding to the integrin alpha 6 beta 1. All right, we don't want more than 1 sperm in there, more than 1 sperm nucleus. So the fast block is something that occurs very quickly. As a response to this receptor protein binding, sodium channels in the plasma membrane of the egg open up, and the secondary oocyte open up, and within a couple of seconds. So when sodium channels open, which way does the sodium ions flow? Into the cell or out of the cell? Into the cell. So that creates a spike, just like depolarization in a neuron. That creates a positive spike of current. And that electrical current flowing across the egg cell membrane prevents other sperm that might have penetrated the zona pellucida from binding to other integrin proteins. All right? So that happens quickly. And then as a response to this current, thinking back to BIOS 430, as an action potential traveled down the axon of the nerve and reached the synaptic bouton, opening sodium channels in the synaptic bouton and continuing that current. You also have calcium channels in the synaptic bouton. Remember that? Calcium flows into the synaptic bouton and affects a molecule called syntagmin. And that allows the synaptic vesicle that has neurotransmitters to fuse with the membrane and open up. So what we're seeing is calcium channels open. Calcium flows into the nerve, and vesicles containing the neurotransmitter, now because of the presence of calcium and its effect on other proteins, are able to fuse and empty the neurotransmitters out of the cell. So we already have that relationship between calcium ions and the fusion of these membranes. Okay? So that leads this fast block to polyspermy, creating this current. That current travels through the cell, and as it does so, it reaches structures that are similar to the sarcoplasmic reticulum of muscle cells. What kind of ions are contained in the sarcoplasmic reticulum of muscle cells?
Calcium.
Calcium ions. So in the egg, that current, that sodium ion, causes those channels in these sacs containing calcium ions to release. The calcium is released into the cytoplasm of the egg. Here it's coming from outside the cell, but in the egg it's coming from inside the cell, like a muscle cell. And those calcium ions are released, and they increase the probability that granules just under the plasma membrane of the egg can fuse with that membrane. And these granules cause the zona pellucida to become cross-linked and hardened, to separate slightly from the plasma membrane of the egg, and to destroy the proteins here that are binding with the receptors, so sperm can no longer bind to the zona pellucida. So we have two mechanisms for preventing sperm from entering the egg. The quick one stops sperm from actually binding to the egg membrane itself. The slow one stops sperm from binding to the zona pellucida, or being able to get through the zona pellucida. All right?
Did you say the zona separates from the...
It pulls away a little bit. You'll see that in our sea urchin. Instead of a zona pellucida, they have a thin membrane, and you'll see that occur. This diagram here, the patella membrane, is what we see in the sea urchin rather than a zona pellucida.
What are the sacs in the cytoplasm that release the
They
calcium?
contain enzymes and other proteins that change, cross-link the proteins of the zona pellucida. So the little sacs are called cortical granules. They don't contain the calcium ions, but because of the release of the calcium ions, they're able to merge and fuse with the plasma membrane. So I think I can erase this cell here. So if I diagram that out, here would be the plasma membrane of the egg. Here would be the zona pellucida. And here would be our first sperm. Okay. So the first step is the fast block. And that is the opening of sodium channels. So sodium ions flow into the cell, okay? And that changes the ability of additional sperm that have already might have made it through the zona pellucida to bind to the egg membrane. Then these ions, these sodium ions, will affect vesicles inside the cell that have calcium. So the sodium ions crossing that membrane will cause these calcium channels to open. calcium ions will be released. And let's find a different color here. Calcium ions
That's already the second block, right?
now this is the slow block here. Calcium ions then would change the membrane characteristics of these cortical granules and allow them to fuse with the plasma membrane and release their contents into the space between the egg and the zona pellucida, cause a separation, slight separation, and hardening, if you will, of the zona pellucida. So that prevents any other sperm from making their way through the zona pellucida, and that's the slow block.
So in the fast block,
Uh-huh.
when the sodium enters into the egg, it blocks the sperm from binding to what?
So additional sperm that might have gotten through the zona pellucida, right here, won't be able to do this. They won't be able to bind to the plasma membrane of the egg.
Okay.
The slow block prevents any additional sperm from getting through the zona pellucida.
So the integrin alpha 6-beta-1 protein is the receptor.
It's on the egg. Yeah, it's the interaction, so the sperm and the egg have the proteins to interact with each other. And the integrin alpha 6-beta-1